Hello, everyone. Welcome to the JCU TropWATER webinar on sea turtles. My name is Chris Kleger. I'm the lead for the Marine Megafauna Group at TropWATER JCU in Townsville. I just wanted to start off by acknowledging the traditional owners of the lands and sea country which we are joining today. In Townsville, we acknowledge the Wulgurukaba and Bindal people. And in Cairns, we acknowledge the Irukandji people. And we pay our respect to elders past and present and extend that respect to all Aboriginal and Torres Strait Islander people joining us today. So this session is going to be recorded and it will be available afterwards on the TropWATER website. The way it's going to go is there's going to be a Q&A session after our key presenters have presented. So at the end of the session, we really encourage you for you to use the chat function at any time to add your questions. And if you'd like to unmute to ask a question during the Q&A, please raise your hand, use the raise hand function, and you'll be invited to unmute. And there'll be more details on how to do that in the chat. At the end of the event, you'll be directed to a quick survey. And we'd really appreciate it if your feedback could be put in there to improve our future TropWATER events. Okay, so that's all of the admin stuff gone. Just wanted, I guess I'm very honored to be here. I'm privileged to introduce you to Dr. Emily Webster and Dr. Caitlin Smith, both JCU TropWATER researchers and turtle specialists and part of this Marine Megafauna Group at TropWATER. Quickly, a quick bio on Emily and Caitlin. So Emily is a quantitative applied ecologist, which means she's making data useful for people and policymakers to help improve the health of the natural environment. She's interested in wildlife and their habitats, especially spatial and temporal patterns. And Emily's PhD was about movement of the green turtles in foraging grounds in Queensland. Why do they stay? Why do they don't stay in places? And what this tells us about the health of their foraging habitats. I know also that Emily is concerned about climate change and what scientists can do to make a meaningful contribution to this complex issue. So welcome and thank you for being here, Emily. Caitlin is going to come second, and Caitlin is a marine ecologist with a background in climate change, marine debris, and ecotoxicology, with a focus on understanding how human-driven pressures shape marine ecosystems. Her work is strongly applied, aiming to generate evidence that supports practical management decisions and delivers tangible conservation outcomes. Caitlin is particularly interested in how cumulative stresses affect long-lived species such as marine turtles, and how science can be translated into meaningful actions for both ecosystems and communities. So thank you both for being here. I'll leave the floor to you, Emily, to start off.
All right. Hi, everyone. I'll just share my screen and get this thing started. Okay. All right. I'd also like to start by acknowledging that I'm presenting from Bindal country. I recognize that I'm privileged. I've had access to a formal education, the opportunity to pursue a career as a scientist, and the platform for my work to be taken seriously in informing ecological knowledge and management practices. But for many other people, the knowledge systems they hold are often less recognized or valued, despite their importance in understanding and caring for country. So I just wanted to acknowledge that Australia's first peoples were its first scientists and the first carers of its lands and seas. And this work is by artist Robert Paul, who's a Juru man, and it represents the coming together of two knowledge systems. So we have the blue figures, which have 3 dots in front of them representing scientists and their technology screens and data that drive their knowledge. And also in the red, we have our traditional owners with the 3 lines representing cultural, physical, spiritual, and intergenerational knowings from their connection to country. And I'll come back to this concept at the end of my talk. So today we're talking about turtles. I just wanted to start by looking into why turtles actually matter to us as humans. So in the GBR region, turtles are a significant cultural species for many First Nations cultures. They appear in some of the ancient songlines. They are totemic, and there are many traditional hunting practices which use turtles for food and ceremony. They also draw a lot of attention and economic revenue to our region through tourism because they're charismatic and iconic. They play an ecological role in our seagrass and coral reef systems through food webs, through ecosystem engineering, and also through nutrient transport, even between our marine and our terrestrial environments. And also importantly, they're often used as a flagship species for conservation. So that means that they can draw attention to broader issues because people are sort of drawn to them and they are such charismatic animals. So most famously, the turtles are used to raise awareness about plastic pollution in the marine environment. But the reason that I study and care about turtles is because they are what we call sentinels of the sea. And this concept of sentinels is essentially species that respond to and reflect changes in the ecosystem structure and function that we would otherwise be not able to observe directly. So that means that by observing turtles, understanding how they use their habitats, their behavior, their physiology, all of this reflects the condition of the whole ecosystem and how it might be changing over time. And some of the big changes that we're seeing these days are around the issue of climate change. And for marine turtles, what we see in the climate change space, especially in nesting grounds, are things like feminization. So turtles have what we call temperature dependent sex determination, and that means the temperature of the nest or the sand affects how many females and males and the ratio of the sexes are born. And we say cool dudes and hot chicks. So that means the hotter it gets, the more likely you're going to get a female biased population. And for many populations across the world, the proportion of females is increasing. If temperatures become too hot, it can actually cause total egg mortality for nests. And in Queensland, we get our nesting occurring sort of between the November to March period, with peak temperatures occurring in January and February. Heat can also affect the number of hatchlings that make it through that incubation period and their fitness. And Caitlin is going to talk about that concept in a lot more detail. In Queensland, we're seeing also issues with severe weather events. So sometimes nests get totally eroded away or flooded. And in some locations, hot and dry conditions are making it very difficult for females to dig their nests because the sand caves in. And at Rain Island, we also see females who become exhausted and die because they're unable to move around in the dry sand. And this led us to try to start looking at this question of nesting refugia, which essentially are sites where we expect that the future exposure to climate change is going to remain stable or remain viable. And so this is a project where we developed a collaboration with the federal government and with the Sunshine Coast Regional Council. And we were essentially trying to leverage free and publicly available data sets to try to develop this analysis to identify nesting refugia. And in particular, we were interested in the Southwest Pacific loggerhead turtle, which is a genetically distinct population. And loggerheads are listed as endangered under national and state legislation. And they have historically seen declines in the population numbers. So this graph on the right-hand side shows the number of nesting females that were recorded along the Woongarra coast, which is one of the sort of densest nesting areas for this population. And around the mid-80s to the early 2000s, we saw declines in those numbers, and that was associated with bycatch in trawl fishes. And then in the early 2000s, we had the introduction of mandatory turtle excluder devices in trawl fisheries, and we can see some recovery of those numbers since then. But that recovery hasn't been ubiquitous across the whole population's range. And in fact, on our GBR islands, we're not seeing that recovery so strongly. In addition to all of this, The mainland nests of these turtles are often predated upon by feral species, and we have some sites that are very low in their hatchling productions, and sometimes this may be due to excessively hot sand temperatures. And so for this study, we were interested in identifying potential nesting refugia. So as I mentioned, those are areas whose exposure to climatic disturbances is predicted to remain stable over time. We also tried to look at potential areas lost, so those that may no longer be viable in the future. And all of this was about working towards defensible procedures for prioritizing nesting sites across the population range. And this whole concept and this work is stipulated as aims and objectives at multiple management scales. So in international, national, state, and local species management planning documents. The first step of this was to establish the nesting range for the species. So we looked at historical data from the Queensland government and from the New South Wales government to identify sandy beaches contiguous with locations where loggerheads have nested in the past. And then we adapted approach from CSIRO, from Adapt NRM, which essentially is based on this idea of collecting climate data at a baseline and then calculating the change from that to a future time point. And so we looked at the 2050 decade centered around 2050 and around 2090. And we looked at sea level rise, temperature, tides and storm surge, and significant wave height under the SSP 585 scenario, which is considered to be our sort of worst case climate future. And we also calculated a net exposure metric, which was the sum of all of these different variables. And we said that refugia would be any place where that net exposure is close to zero. We also collected a lot of physical characteristic information. So we wanted to know given the different climate exposure, which sites would be more or less susceptible to those climate impacts. And so we were looking at historical coastline change, coastal squeeze, so the distance between the beach and structures, man-made structures in the landward direction. We looked at beach slope, vegetation, and coastal armoring features as well. And what we found was that none of our sites had a net exposure metric close to zero. So everything was greater than 1, which means that essentially all the sites are projected to experience increased climate exposure. That change in exposure was the highest in southeast Queensland, so from Gari to the Sunshine Coast, Minjerribah and Mulgumpen. It was also high in the Swains and the Capricorn Bunker groups of islands, and Tweed Heads, Grafton and Taree in New South Wales. And we had the least change in exposure in southern New South Wales, between Brunswick Heads and Esk, and at South Island, Northwest Island and north of Moreton Island. And then when we break down that net exposure into the different components, we saw that we are getting, as we said, increases across the range. So the minimum increase in temperature that we saw in the 2090 future is 2.58 degrees. In terms of sea level rise, we're getting the greatest increases around the Swains, Gari, and Rainbow Beach, and south of Barkundjia. I think that's how that's pronounced, but I'm not sure. And then in terms of damaging waves, we are getting the greatest exposure in the northern part of the nesting range, including the GBR Islands. We also had a little bit of a look at potential suitable areas in the future. And the way that we got at this was we looked at our historical nesting data, and we found that those historical nests occurred between temperatures of around 22 to 27 degrees on average. Those are average summer temperatures. And then if we project into the future, we could see that that mean summer temperature exceeds that suitable range for nesting in the areas that are north of around Agnes Water and Woodgate, if we look at 2056 to 2085. And then if we look even further into the future, to the decade centered around 2090, most of the Queensland nesting sites exceeded that suitable temperature range for this stock. In terms of susceptibility, so this map on the left shows the distance to landward structures that are within 300 meters of the beach, and some of the areas that flagged as having a risk in terms of coastal squeeze were those areas with structures close to the beach, including the Sunshine Coast. We also had evidence of eroding shoreline, so that's the reddish colors in the middle panel, and that was evident along the Capricorn Coast, among other sites. And the Capricorn Coast is important because it's one of the dense nesting areas for this stock. And then we had a master's intern who came and did a lot of island change work for us using remotely sensed images. And what we found was consistent with other work in this region, which shows that those small, elongate, unvegetated coral cays are the ones that are especially vulnerable to change. And this was particularly interesting for the Swains reefs because they're highly dynamic and there are very few alternative nesting sites nearby the Swains. I guess one light at the end of this dreary dark tunnel is that we are seeing some examples of turtles responding to this future change. So, for example, there are some populations that are exhibiting phenological plasticity in their timing of breeding and also the location of breeding. And so this image here is from a loggerhead that we tracked from Wreck Island. And we can see that these Southwest Pacific loggerheads, through tracking studies, have quite plastic site choice in terms of nesting, and that is at a scale of sort of tens to a few to tens of kilometers. So this would indicate that there is some potential resilience built into this system, and turtles may choose to nest at several different locations that have different amounts of viability as long as those are relatively close to each other. So future directions for some of this work is that we have recommended to integrate these results into spatial risk decision matrices, which we will hope to develop with our partners. And this will be used to prioritize nesting sites for protection, monitoring, and climate adaptation actions. We're hoping that in the future we can develop similar frameworks for other threatened species populations or marine turtle life stages, including foraging grounds. And this work is informative for evaluating the scalability, suitability, and the cost benefit of different interventions. And Caitlin will talk about that more shortly. But before I pass on to Caitlin, I just wanted to also do a little plug for Dugong Connections. So this year and last year, I was fortunate to be part of this project, which is all about aiming to cultivate genuine connections between scientists and traditional owners. So it's about sharing contemporary Western science and also discussing opportunities to work together and how to do this in the best way, and providing opportunities for traditional owners to share with us as well. Because dugongs and turtles have similar habitats, they face similar pressures, and they both represent culturally important species. Some of the groups that we've connected with through this project have shown interest in dugong and turtle discussions and activities. And so at the start of Dugong Connections, I did some preliminary work, which was a desktop study, a literature review that was asking questions about the levels of participation of traditional owners in turtle projects in the region, what local knowledge gaps there are about marine turtles, barriers to participation in marine turtle work. And I presented those results at the Australian Marine Science Association conference in Melbourne in 2025. So if anyone's interested in that work, I'm happy to provide more information. And Dugong Connections has also led us to some opportunities to work collaboratively in the field. So in October of last year, we went out to the Whitsunday region, Ngaro country, with Ngaro guys and also the Queensland government, QDETSI. And we were essentially looking for opportunities to work with turtles, and we managed to catch turtles and dugongs. We managed to catch 2 green turtles and track them with GPS. And this activity was sort of a means of us getting on country together, sharing knowledge, and building those relationships. But also nicely, the tracking data from this little study was quite intuitive to interpret and has done a really good job of demonstrating some of the characteristic patterns of behavior that we see in foraging green turtles and how varied these can be. So on the left, we have one large female who was tracked in the intertidal area there in Pioneer Bay and has essentially stayed there for several months and is moving in and out with the tides and foraging on seagrass. And we also have another individual on the right, also a female, who has demonstrated that green turtles are capable of very long distance movements. So she's moved all the way down to Shoalwater Bay. And this just shows that our wildlife are connecting our seascape across very long distances. But thank you very much. And with that, I'll pass over to Caitlin.
Oh, thanks, Emily. It's such a hard act to follow. It was fantastic. Okay. Okay. All right. Good afternoon, everyone. Thank you so much for being with us today. My name's Caitlin, and today I'm going to be talking to you about a project called Healthy Hatchlings. And this is a project that's a collaboration between Griffith University and TropWATER at JCU. And funded by SeaWorld Foundation. I'd also like to thank my collaborators, Kim and Jason from Griffith, for all of their help throughout this project as well. I'd also like to acknowledge that I'm connecting from the lands of the Irukandji people today. I pay my respects to all elders past and present and any traditional owners that are joining us today. I'd also like to acknowledge the Port Curtis Coral Coast Group, which includes the Tarabalang Bunda, Gureng Gureng, Gurang, and the Bailai peoples where this field work took place. So Emily already kind of introduced some of the problems that our turtles are facing. They really get hammered by a lot of human-based impacts, and this is exacerbated along our coastlines, particularly in the nesting phase of their life cycle. So we see increased coastal urbanization, and climate change is increasing the intensity and severity of cyclones. We also have sea level rise, erosion, and then we also have our increased sand temperature from climate change as well. And then in addition to this, we also have things like predation. So, and these can be from both invasive and native species. So, particularly up north, we have our feral pigs and foxes. And then we also have things like ghost crabs and goannas as well that like to eat sea turtle eggs. Next slide. Oh, okay, hang on. Sorry. There we go. Cool. And so one of the main reasons why scientists are concerned about climate change, particularly for sea turtle species, is like Emily said, is the display temperature dependent sex determination, which means that the temperature of the sand dictates whether or not the hatchlings are male and female. And so like Emily said, hot chicks and cool dudes. So higher temperatures is female, lower temperatures are male. And so this is really causing a shift in our primary sex ratios, and particularly for some genetic stock like the northern Great Barrier Reef green turtle, we're seeing a really extreme female hatchling bias. And we also have things like pivotal temperatures. And so this is the temperature at which you theoretically get 50% female and male hatchling production. And this differs greatly between species and genetic stocks as well. But there's also evidence to suggest that there's local adaptations of pivotal temperatures, which could help with the production of males at warmer sites. So we typically see a higher pivotal temperature at more equatorial sites and warmer sites. And so there are a lot of existing management solutions, particularly for our nesting beaches, depending on what issues you're facing. So things like irrigation, whether or not that be seawater or freshwater, and also shade structures. So hatcheries, you can have shade cloth, or you can also use palm fronds. But these management solutions typically mitigate against increased sand temperatures. And then you can also have relocation as well. And so when we talk about doomed clutches, we're talking about clutches that would typically not develop because they're at risk of being inundated by seawater or erosion, or there's a high predation risk in the area from pigs or foxes, so they get relocated into predator exclusion cages or higher up the dune. And particularly for our management solutions, when we're talking about irrigation or shaded hatcheries, the aim of this is to move those sand temperatures into a male producing zone, and they have been quite effective at doing that at multiple locations. But there are also a lot of unintended impacts from these management solutions. For example, we were trialing irrigation a number of years ago, and you can see this graph down the bottom left-hand corner. This is where we were trialing an irrigation protocol, and we actually ended up increasing sand temperature. And this was because when we watered in the early morning, the water was sitting at the top, and then as the sun came up, it heated that water up and then percolated to nest depth, increasing the sand temperature at nest depth. So had the exact opposite effect of what we were anticipating for that. Then we also have the shifting of sex ratios when you're already a male producing area as well. And I think this happened at a couple of places where we had these papers come out and say, yeah, we've got this feminization issue, and people were like, oh my God, we don't want that happening to our beach. So let's put up a shaded hatchery and start relocating into there without knowing the full picture of, their sex ratio of their beach as well. And we also have a spatial variability across beaches and islands where like one side of the island will be a female producing side and one side being a male side. So knowing your spatial variability and your hatchling output is also incredibly important in that. And also increasing your male hatchling production has been shown to have long-term population decline because you're reducing the number of nesting females as well in doing that. And this is a really complex system. So yeah, I love turtles. They're great, but I think what more so I love is I love a challenge. And so changing one variable in this very large puzzle can have so many flow-on impacts. And I've been told many times that I have a face like this, and it's probably because I'm thinking about this problem and all of these variables as well. So we need to know so many things prior to implementing these management methods, such as what your hatchling sex ratios were in the past, what are they now, what are they going to be under future climate scenarios. Also things like, what are your current pivotal temperatures for your genetic stock, and has this changed over time? And then also, how do your sex ratios change over your genetic stock or even spatially across your island or beach? Then where do your nesting females breed? And then what are those operational sex ratios at those breeding areas? And finally, what are your male remigration intervals? Because we found that historically a more female sex ratio in your operational sex ratio is more beneficial because your males have a shorter remigration interval between breeding seasons. And then finally, how do all of these variables change over space and time? So yeah, if you ever see me looking like this, it's probably because I'm thinking about all these questions to answer. And so that brings us to Healthy Hatchlings. So like I said, it's a collaboration between myself and Jason and Kimmett Griffith, and funded by SeaWorld Foundation. And our goal was just to look at one small piece of this puzzle. So look at the implications of our current management solutions, whether that be relocation, shaded hatcheries, or our predator exclusion devices, and look at the sublethal impacts of hatchling health and locomotor performance, because we want to be producing the best quality hatchlings to increase survivorship, to boost our populations. And the final thing for this is to produce recommendations for protocol updates and potentially have a management decision tree or management matrix as well. So our first point of contact is we went to Monopro Conservation Park in December. Me and my master's student, Maddie, shout out Maddie if you're here. And we used 40 loggerhead clutches in total. We relocated 30 of these, and then we left 10 in situ. And all of these had hobo temperature loggers in each clutch. So this is our experiment site. So like I said, we had 10 clutches left in situ. Then we relocated 10 into the predator exclusion cage, 10 into the shaded hatchery, and then we relocated 10 up into that bare dune area as well. And then we came back in January and February of this year, and we put out our little mesh guard cages around each of our clutches. And it was my volunteers checked the clutches every half hour to an hour throughout the night. And if any hatchlings had emerged, we then took 20 of those to weigh and measure and also look at any scale mutations or abnormalities in these hatchlings. And then we put them through what I like to call the little hatchling Olympics. So we looked at 3 energetic or locomotive performance tests. We did self-riding. So hatchlings are the only stage of a sea turtle's life where they're able to flip themselves over from their back. And this is a really good indicator of how energetically well they're performing as a hatchling. And then we also have our crawl and our swim speed as well. So we have these little raceways either filled with sand or seawater, and we timed how quickly they were crawling and swimming as well. And then it's everybody's favorite thing to do, nest sticks. So we also looked at hatching success. So a couple of days after the majority of the clutch had emerged, we go back and we dig up the egg mass and look at how many eggs had hatched. And then it's pretty gross. Some of the eggs have been in there for like 2 months, just rotting away. So thank you to my volunteers that helped me do this. For the eggs that are unhatched, we do embryonic mortality staging. So we look at these 6 phases here on the right and see at what stage the embryo died. So because we only came back from the field about 3 weeks ago, these are very preliminary results. So I was like furiously writing some R script last night. But first up, we've got our hatching success, and we found that there wasn't any significant statistical significance between treatments in hatching success. However, our bare dune treatment here on the left had a lot of variation in success and did have lower success in comparison to the other treatments. However, it wasn't statistically significant. But our clutches that were left in situ had the highest mean success as well. And to me, I also thought, okay, well, if hatch success, hasn't changed between treatments, maybe something in our embryonic development can tell us like what else is happening throughout development. And there also wasn't any significance between embryonic death throughout incubation as well between the treatments. However, you can see here that the hatchery did have a lower later incubation mortality, and you can kind of tell by these temperature traces. So these are the temperature traces for the 4 treatments throughout development. So at the top here, we've got bare dune, and then control, predator cage, and the hatchery. And the clutches in the hatchery had a much longer incubation time due to lower temperature. So in the same breath as saying sand temperature influences sex, it also influences your incubation time as well. So a higher temperature means a shorter incubation time, and a lower temperature means a longer incubation time as well. And then along here, we've got our pivotal temperature for the genetic stock as well. And you can see that the majority of the clutches in the bear dune, the control, and the predator cage were above this pivotal temperature for the majority of um, development. And then your hatchery is below this. And these two lines here are the average thermal sensitive period. So this is typically where we see our gonads differentiate between male and female sex organs. But this also shifts based off your incubation period as well, but this is just an average. So you can see here that the hatchery was below the pivotal temperature at some point during the thermal sensitive period, and the predator cage also kind of dipped below this line as well. And I guess that's also why, and if I go back, we see like maybe a little bit of a dip in late stage embryonic death is because like towards the end of development, when you've got really high metabolic heating, that the clutches that aren't so heavily shaded have like these really high nest temperatures, whereas the hatchery had these lower temperatures. There was a bit of rainfall here towards development and also right before the thermal sensitive period as well. So I'm so sorry, I haven't had time to analyze the locomotor performance of the morphology, which is also the really cool stuff. So really watch this space for that, and also recommendations for existing protocols. Our next steps are to take this information and kind of go a bit wider with it and have workshops with turtle monitoring groups to create tailored management plans based off their concerns for their particular nesting stock. And really try and understand what data is required to make these informed decisions. Do we need to put out more temperature data loggers? Do we need to do more updating existing hatchling sex ratios to get a more current view of population trends as well? I just really love to acknowledge a few people. So particularly our volunteers, Tristan, Ariel, Maddie, Meg, Nicole, Lucy, Nikki, Jacob, Dylan, and Ella. Thank you so much for all of your help. Again, a big shout out to Jason and Kim at Griffith and SeaWorld Foundation for their funding support, the TropWATER team. A big shout out to the Depsy team, Kyle, Michael, Abby, Owen, Maggie, and Julian, and Gus at Stylidia for creating like the most beautiful pictures and videos as well. So thank you very much. And here's my email if anybody needs to ask a question that we don't have time for today.